So the first presentation of the second half of the morning session is an area close to my heart, GPCR drug discovery. A very warm welcome to Alistair Brown from Heptares, who's going to give us a run-through of structure-based drug design as it relates to GPCRs and how their technology really opened up hit ID opportunities at this important target class.
So, thank you very much, Alan, and quick thank you to the organisers and the sponsors of this for making the meeting happen. So, I work at Heptares. We're a company focused on GPCR-based drug discovery, and I'll give you a sort of whistle-stop tour through some of the things we've been developing and leveraging in that space to help drug development. So, we heard a little bit earlier, actually, from Lawrence about GPCRs, and that's one of the superfamilies which has been targeted historically for a long time. I guess it's sort of, perhaps interest, like most things in life and certainly in science, goes in cycles, and the cycles of GPCRs in large pharma are sort of decreasing at the moment. Now, I'll try and argue that shouldn't be the case, and that there are plenty of good targets there. I think we've picked off a lot of the low-hanging fruit, and perhaps there's a lot left which are intractable, difficult to target for various reasons, be it selectivity, compound properties of compounds that are found to hit it, or just general intractability. So we're all very aware of things like GLP-1. Orexin 2, for example, for narcolepsy, which is, has quite a strong genetic validation behind it, but yet has still been intractable in terms of targets. And compounds like CGRP antagonists, which made it to the clinic, but actually their properties are horrific. They've been very difficult to target. They've led to idiosyncratic tox mechanisms as well. So, there are plenty of reasons to go back to the GPCR target space. Not least that it's huge, and there are a very large number of targets in there which still remain to be mined effectively. So, being Manchester, I couldn't resist a small analogy. I think GPCRs are a little bit like United. There's plenty of opportunities. It just needs the right way to put it all together, and I'm not entirely sure that was it. So what I'll argue is there is a different way for GPCRs. It's not the historical HTS, and that structure-based design can be used to pick off some of those more challenging targets. So Heptares, we are a biotech. We're focusing on GPCRs. We're what I call a sort of early clinical biotech now. We've got our own program in phase 1 and a clinically ready IND compound, which is also our own, which is ready to go into phase 1, along with an emerging portfolio in the neuroscience and the metabolic disease area. And alongside that, we run a lot of partnerships with large companies as well, and Lawrence touched on something that we've been running with AZ, and that seems to have been quite successful. What we've done really is pick out a niche, and the niche really comes very much from what's led the way in terms of enzyme-based, structure-based design, and how people have leveraged that to design more selective, more tractable outcomes for drugs, for enzymes. But in GPCRs, clearly there's a number of missing links in that, in the ability to produce a lot of stable protein in the form that's suitable for crystallization, and biophysical approaches. But we also heard about the value from a screening perspective in confirming hits in different formats. And that's really where the Heptares approach is targeted. So we use this idea on this concept of a stabilized receptor to introduce specific mutations well away from the binding sites to stabilize the protein and enable high-level expression in both insect and mammalian systems. And that can lead and can form the basis of computational-based structural information, high-throughput screening in a sort of fragment-based setting, and then ultimately structure-based design. It looks linear in this sense. It's not necessarily always linear, and we try and parallel a number of these activities where possible. So the star is the sort of the basis that underpins everything that we do. So stabilized receptors, and everyone is fully aware that GPCRs have multiple conformations, and it's those multiple conformations which make them so challenging to overexpress in a membrane environment, where when you begin to remove the membrane, which you need to do for the purification process, the conformational flexibility precludes them from really crystallizing out. So by introducing specific mutations, and in this case for the adenosine A2A receptor, there were 4 specific ones which were introduced, you can create a far more stable protein which doesn't require a lipid environment around it to hold that conformation, but is much more stable in a minimal lipid environment and suitable therefore for sort of Xray and biophysical type of approaches. Now, of course, we validate the pharmacology, and here you can see for this receptor, we're comparing the affinity of the wild type to the star, and you can see there's absolutely no change in the pharmacology associated with any ligands that we tested at that receptor. And actually, we'll come back to the pharmacology, because it's not just there aren't differences, there can be created differences, and you can even use that to your advantage to screen certain conformations of receptors. I think that touches on a very exciting emerging area of sort of bias signaling, which has been there, and again is one of these circles which I think is coming back up to the forefront. This is, I think that, the other key piece to really highlight here is that it is ultimately transferable across the entire superfamily. So, we've now published on and have internal examples from class A, B, and C GPCRs where they're all amenable to this sort of approach in both agonist and antagonist format. So this isn't a sort of, a one-off. It seems to be suitable and amenable for every receptor. Clearly, they have to be tailored to the specific ones. It does take a little bit of time, so this is not a quick win necessarily, but this is about picking the right targets to do these sorts of approaches on to give you the right chances of success clinically. So this is the sort of basic process, I suppose. So we start with an unstable GPCR. We use a ligand to try and stabilise the receptor in a conformation, although it can be done in a ligand-independent manner. Introduce a number of mutations, and you can see here, it's up to 1,000 or more, both individually and in combination. And in each part of that process, those are then analysed for the stability of that protein. So we use thermostability assays that look at the denaturation, so the unfolding of the protein, and then monitor that as we increase the stability. And so typically we're looking for a sort of 40-degree stable in a minimal detergent as an outcome that's then would be what we would call suitable, or a final star, and suitable for crystallization. Along the way, of course, the stars can be sort of iteratively used for other processes. They don't all have to be fully stable for the final Xray, and they can form part of drug discovery programs at an earlier stage as well. So here, we can see we monitor the increase in stability of the protein from the wild type to the star. We monitor the pharmacology, in this case in the inactive state. And so this is an antagonist star for the adenosine A2A receptor, where the antagonists maintain their affinity, but actually the agonists drop off and lose their affinity. So you can see here we're sort of, gives you a hint that this is stabilising in a conformation which is not amenable to agonists, but is amenable to antagonists. And of course you can switch it. And then, a number of other techniques are used to monitor the quality of the protein alongside this sort of iterative process, ultimately leading to the opportunity for structures. So this is, as I said, is not just a one-off. It's, we have examples across the entire superfamily now where stars have been made and are suitable, demonstrated properties that would be consistent with crystallization. We haven't crystallized them all. I think we've generated maybe greater than about 35 crystal structures now in-house from 17 different backgrounds. So, clearly not all of that is published. We'll touch on a little bit of that today. A number of those are proprietary. But the point being that it's very amenable, and it does work, universally across the superfamily. So here's just a few examples. I doubt you can read them all, but you can see in each case the wild type is in red, and the consequence of the stabilization is in green in terms of the stable receptor. So you can see some wild type receptors are very stable originally, and the star doesn't really add a lot more stability. It can help the expression and purification, however, when you begin to truncate them, it becomes even more important to have that. But other receptors are very, very unstable, even in the wild type form. So, it's what you would expect from a superfamily in this sense, I suppose. They have evolved their own specific requirements for stability, and I think there's probably a lot more that underpins why they've done that. We haven't really got into that space. So G-protein coupling and specific G-protein cellular regions, lipid environments, and those sorts of things. But what's clear here, I hope, is that in every case we can introduce a number, and a very limited number of mutations, which in each case will not influence the pharmacology of the ligands that we know about, but will create a stable receptor suitable for a number of other approaches. And it's those sort of approaches that I sort of like to spend most of the rest of the talk going through, is what we do with those stabilised receptors. So these are the sorts of, this is just a nice picture of some of the crystals. You can see they're not all the same. We've got a number of targets. We capture these nice pretty pictures, as well as, of course, interrogate them at a molecular level. So what can we use these for? And they've talked about the star. This is, again, I'll stress this again, this is a sort of, that underpins everything we do. So we can use it for structural work, and I'll come back and talk a little bit more about some of the structural stuff. But I'd like to spend a few minutes on fragment screening and the approaches we use for that. A few minutes on the sort of conformational side of this, and why and the consequences of stabilisation, and then think a little bit more about some of the computational and sort of biocore studies that having a stabilised receptor that you can take out of the lipid environment, and so do traditional SPR type approaches on, really offers. So there's a little bit, I think Lawrence talked earlier about fragment screening and the opportunities there. We really very much believe in fragment screening. at Heptares. And I think that, the STAR process that we work through gives us access to protein in both membranes, which is very stable, expresses at a high level, is suitable for working at much higher DMSO concentrations than would normally be usable. So we have a wild-type receptor that we can screen on, but we also have a stabilized receptor that can be taken out of the membrane environment. And so it's amenable to all of the biophysical approaches which you've heard about from other speakers. So, NMR, SPR, TINs, high concentration screening, capillary electrophoresis. They're all amenable to this sort of to the stabilized protein in solution. And so that gives us this opportunity to really interrogate proteins in terms of small binding sites, and then to use the structural information to leverage the rapid optimization of those. So we use these sort of spider diagrams to sort of plot the properties, and the further away from the center you are, the less good the properties are. So you look at lipophilicity, selectivity, C log P, blah, blah. So this is a starting point, for example, that we may have found from a high concentration fragment screen, and this is where we'd like to end up, as close to the center as possible. And I think in almost every case across our programs, we've been able to take either historical compounds or our own leads and show a very, very clear optimization based on our structure-based and computational approaches. This is an example here for the beta-adrenergic receptor that was published recently in the JMEG Chem, where we took a compound with suboptimal properties that had very weak affinity, and within very few rounds had been able to identify a sub-nanomolar compound. So I spoke a few minutes ago about the sort of conformational landscape that the receptors that the STAR generate offers as an opportunity. And so we did quite a significant amount of work in the early stage of the company on the adenosine A2A receptor. And this has been published recently in Morpharm. You may have picked up on this. But I think the thing I'd like to really stress is that in creating a STAR in either the agonist or the antagonist state, we're able to selectively focus or increase the affinity for different pharmacological profiles. So here is a map which is really looking at the difference between the stability and the affinity of the protein in the active and in the inactive state. And you can see, so this is an agonist, NECA. This is caffeine, which you've just had in your coffee as a sort of neutral in this case, and going all the way to an inverse agonist on this side of the spectrum. And you can see here, so it's a bit complicated really, but what this is really saying is that the agonist NECA has lost affinity to the inactive or the antagonist star whilst maintaining affinity in the agonist star. And at the other end of the spectrum, the inverse agonist has maintained its affinity or increased affinity at the inverse agonist star, but lost affinity significantly at the agonist star. So what we're doing here is tailoring the conformation specifically to the properties or to the pharmacology of the ligands. So this holds true for the principle here can be defined or can be proven from a cellular-based mechanism as well. So in a sort of a conditional overexpressing system where you can titrate in a level of constitutive activity, you can prove in the same setting that these are agonists. These are as neutral as you can see in the setting, and these are inverse agonists. And then if we put that into the context of the structure, so we also published the structure from these ligands, along with another group at the same time. And so here you can see there's both NECA in green and caffeine in pink. So caffeine we'd sort of call as a neutral antagonist, or NECA as the agonist. And in grey is really, as defined by the crystal structure, the agonist binding pocket available for binding. So you can see both of these fit in the agonist pocket. They make interactions with a histidine and a serine in TM7 and an asparagine in TM5, and have the ability to really activate the receptor. So you get the sort of shift up in TMs 1, 5, and 7, a rotamer toggles around TM6 as well. And that really, it's those shifts which define this cavity here. But for the inverse agonist, you can see, although these interactions are still feasible, it just does not fit in the pocket available within the agonist space. And this fits very much with the sort of pharmacology we see. So in the inverse agonist star, then this pocket is defined out this way. TM5 bulges out and accommodates this extra sort of trail or chain of this, and this is the ZM2413 at 5. So this is sort of how we're beginning to use this structured information to sort of create stars with different pharmacologies, to place those pharmacologies upon both agonist and antagonist structures to understand those interactions, and then use those to really drive the SAR and the optimization of compounds as we move through. And we'll come back to this story of adenosine a couple of times so you can see a few more pieces of work that we've done in leveraging this information. So The other area that having stars and structures has really enabled, and we're spending a lot of focus on at the moment within Heptares, are the sort of techniques we might use in enabling structure-based design. And here I'm talking much more about sort of the SPR-based biophysical type of approaches, but also very much computational approaches. So we use this process called biophysical mapping. I'll just talk a little bit about that in a second. But also, you know, what we're really trying to now understand are more refined, more detailed homology models based on an understanding of waters, where they are and what their properties are. And then also, you know, a number of different grid maps that help us fit into a pocket specific ligands, understand where they may be breaking surfaces that may not be necessarily ideal, and so how that might drive optimization of compound properties. So biophysical mapping is really, I suppose it comes out as part of the process of the star process itself. So as I said, we sort of, in making a star, we'll introduce up to 1,000 different mutations, singly and in combination. So that gives a lot of opportunity to use those mutations to generate further information to understand the protein and its structure as we're moving towards a structure, but ahead of actually getting the structure. And that's really where biophysical mapping comes in. So by picking out specific mutations that we believe are important for an understanding of the binding site, those mutations then can be tested against, in this case, on a biophysical platform. So this is SPR, both looking at affinity, but also looking at the kinetics of the consequences of those ligand changes, and then using that to fall back and refine the homology model ahead of getting the structure, so that we're in a position to enable and to maximize the use of the structure when we get it. So in this case, it was a panel of 8 different mutants against 21 different ligands that were cross-referenced using both affinity and kinetic data to try and refine that homology model and predict roughly the binding site. And you know, when the structure came through, the predictions were really quite close. So, I mean, this is where it led to, and this is sort of part of the idea of the use of the mutant. So here you can see ZM, and we talked about the ZM ligand a few minutes ago with the phenol group, which really extends through a cavity created between TM2 and 7. And some specific residues which were found to be important through the biophysical mapping for the interaction of the ligand. So serine 277, which generally was actually negative in the binding of most of the ligands in the adenosine A2A receptor and the leucine 285. But you can see here that there's quite clearly a different mode of interaction from the triazoline series, which is represented in the cyan colour here. And this is really what we were trying to maximise, so to pick out specific interactions and areas, and this was the sort of the homology modelling and the mutagenesis approach. We'll refine this in a few minutes using water maps as well, to identify specific regions that we may want to target that would offer us a different chemotype, move away from some of the properties of ZM245 that are not necessarily favourable, and then increase the sort of efficiency of the interaction with the receptor. And so on that, you know, I mean, we ended up getting a structure with these compounds in, and so these are the Xray structures with 2 compounds in from that series. So in this case, the compound with 2 methyl groups here with a triazine, is that right? Pyridine, sorry, on that point there. And what this is doing here is making an interaction between an asparagine and then a methionine, sorry, in TMs 5 and 3. And but it does leave open a region here, which is an area that we were trying to sort of exploit as much as possible. And so the introduction of a hydroxyl group there picks up an additional hydrogen bond with a histidine here in TM7, but it also extends slightly the length of these bonds here, and that really enables this to stretch across, make this interaction. You can see in the crystal structure here, this histidine has been just pushed out slightly, enabling it to make an intramolecular hydrogen bond here as well. So this changes the structure of the protein. The consequences of this are a very profound change in off rate as well. So this was picked up in the Biocore assay here, and you can see if we look at the half-life, we move from this compound to a slightly extended half-life to a much more extended half-life here, with a very minimal change in the receptor. So the other area that we've been focusing on and talked about sort of optimizing and suboptimal interactions is the potential for water interactions. And this is an emerging area when it comes to sort of computational insights into where ligands are binding. And we've found these sort of unhappy waters, and generally these seem to link in with a number of the lipophilic groups or lipophilic regions within GPCRs. And it is quite clear that GPCRs do like lipophilic ligands. The question is, where is the best place to introduce, if you're going to take a penalty with that sort of property, where's the best place to introduce that? And here for ZM245, 241385, you can see it does target one of these unhappy waters. These are the ones which, when you seem to hit these regions, and these are, so I had to just backtrack slightly. These come from both predicted water sites, but also as we resolve our X-rays to higher and higher resolution, we see these waters. So we can then both predict and then confirm our predictions, build our models, and iteratively improve our ability to predict it using this approach. So ZM245, clearly hits an unhappy water in this region, but leaves open, an entire region of unhappy waters that one would expect could be targeted, and that's exactly what we tried to do. And as you can see in this triazine compound here, that's really where the shift in affinity comes from the introduction of these methyl groups and nitrogen on that ring system here. And so we're staying and we remain in the same position that the 241385 was at the bottom of the pocket, but have also maximised the value in this region of the pocket here. And so these are the sorts of approaches that we're using to build in to help our lead optimisation processes to build, I suppose, the most efficient ligand possible for the space. And that would really be the key. So I think the one thing that, and I hope at the end of all of this, one message which really comes across is structures, they're incredibly empowering for defining where affinity may come from, where selectivity may come from, and also where efficacy may come from. But the other thing it also gives you is once you have a very, very clear handle on how those properties are being controlled by the interaction with the compound and the receptor, it enables you to take some time to focus on monitoring and addressing physical chemical properties. And that's got to be one of the key areas that a lot of drugs have failed from in the past as well. So if you know where you can maintain affinity and exactly how you're deriving that affinity, you also know then where and how you can modify other parts of the compound to address other physical chemical parameters that might need to be targeted. So, and I think that's a very unique ability of structure-based design. So it's not just about the binding, it's about what that gives you in terms of optimizing other properties of the compounds as well. So I think another nice example, and this is really just a quick snapshot of where water molecules can have other effects. They don't just have to have direct binding effects in the sense of driving affinity, but they can also influence selectivity. So here you see an OX1, OX2 overlay, really, where a single residue here, so alanine 127 in OX1 was, or can be mutated to 3 in OX2. And what this does is change the selectivity profile of the receptor. Now, the ligand EMPA is highly selective for OX2, and only when you mutate the residue can you cross over that selectivity. But what's quite clear is that ligand is actually not making any strong hydrogen bonds with any residues in the entire pocket. What does seem to happen, however, is that the mutation which transfers that selectivity is resulting in a change in the water network in that pocket. So it can be that selectivity can be derived within a pocket that doesn't have any direct hydrogen bond interactions purely by the presence or absence of specific water molecules. And this highlights the importance of these tiny little groups sitting in pockets for GPCR binding. So where we're taking this now is to begin to try and think about how the presence of waters may influence kinetics, and clearly kinetics is something we monitor a lot. We use SPR as an approach consistently through every project. But now we're beginning to think about what the consequences of unhappy and happy waters may be for kinetics. So although this is work in progress, it may or may not end up being right, and I've tried to sort of highlight we have the access to crystal structures to demonstrate exactly where waters are, but also predictive water map tool or water flat tools now, which are being optimized to really identify where these waters may be. But it helps us set up hypotheses about the consequence of this. So in this case, what we're going to test is the concept that unhappy waters may result in compounds which have fast on rates, so affecting the desolvation and solvation properties within the binding site, and that the presence of unhappy waters once a compound is there may influence the ability of those waters to be exchanged, and so influence the off rate of the compound as well. So we can begin now to really formulate concepts for GPCR-based kinetic approaches as well. So really, in summary, I mean, that's how we've been using water networks. The next step, and really where we're trying to help evolve the computational tools, is through molecular dynamics. So here, molecular dynamics are using, from the left here, you see this compound again. We come back to the adenosine. It's not necessarily optimal. By introducing waters in this and releasing a short relaxation period, you can see that there's the presence of these water networks is interacting with the nitrogen on this ring system here. And then ultimately, actually, a continued molecular dynamics for this hydroxyl group on the end here picks up a histidine interaction in the pocket itself. So we're trying to sort of build these processes together. The focus really for us is on multiple properties, so potency, residence, kinetics, and how those link with the water networks in the molecular dynamics. So I thought I'd spend just 2 minutes more tying this up, given that we started a couple of minutes late. Can I get away with that? Going through mGlu-R5, only because I think it's a story that brings it all together. I think it's something that we just published very recently. It's first-class C GPCR, and it adds to the list of amenable targets. So I think you know a lot about mGlu-R5. Antagonists or NAMs have been out there for a while as a potential treatment for autism, migraine, depression. Ross have just published some data recently where it didn't meet the primary endpoints, but the secondary endpoints were quite encouraging for depression. It's a target that we've been interested in. Originally, Adex's compound had very poor PK properties, so we've been, one of the focuses, as I said, was to maintain affinity whilst addressing PK. We've ended up with a compound which has very good properties in terms of once-daily dosing, as compared to an Adex compound which had a very high spike, a great understanding of occupancy. So positioning ourselves to really take this forward now. But really in getting there, we've used the STAR approach to stabilise and to generate the structure of a class C receptor. And I think it's a really interesting example of the binding site for a receptor that would have been very difficult to predict. So this is a metabotropic receptor. It evolved to bind the ligand on the outside and the extracellular venous flytrap domain. So it really doesn't have any reason to maintain evolutionary pressure on the TM domain. And that's exactly how you see it ending up. So TM, I mean, there are some very clear differences. I mean, TM7, for example, has shifted in about 5 angstroms to constrict this pocket and to constrict the entry to the pocket. And TM5, equally, at the top region, has also pushed itself in. And you can see you end up with a very tight binding pocket here that's, unlike other other GPCRs. And really explains why the SAR is so exquisitely tight. And if you look then at mavacluran in this pocket, you can see there are some key hydrogen bonds. But I'd like to bring your attention just to the water compound here. There is a water, we see this in the structure, it's very clearly involved in an intramolecular bridge between TM7 and TM6, so influencing this sort of rotamer toggle concept. And if you take mutations in any of these positions, you can influence the constitutive activity of the system. So just by influencing the potential for that water molecule to be trapped in that state, you can influence that activity. But you can do the same with the compound as well. So you can take a methoxy group here, but change that and increase the bulk and push that water away and then move from NAMs to neutrals to PAMs. So exquisite SAR that can be explained structurally now by the presence of water at the bottom of the pocket. We've approached this from a fragment screening approach. I mean, I touched on the value of fragment screening in terms of stabilising for radioligand binding, and here you can see the consequences of stability for a higher concentration of DMSO. And this really just means we can screen at a higher concentration in a normal radioligand high binding assay, but it can be also used in solution as well. In our sort of bullseye approach, the aim was to move as close to the centre as possible, and we've ended up down there within about a year with this program. So it's enabled us both to optimise properties, maintain affinity, and move quickly towards what we now have as a sort of candidate molecule. So I won't spend any time really on this. I'd just like to highlight there are opportunities for these stabilised proteins once you have them in solution for a number of biological approaches, and clearly they could be used to select antibodies or peptides. As a summary, I'd say they are a very enabling part of what I believe is the future of targeting specific GPCRs. They enable a number of things that, of course, we bring back to the wild-type receptor in every case in the end, and everyone would have to. But they certainly, and I hope I've shown you ways that they enable us to get there in a different way for targets that may well have good clinical validation but very poor molecules. And that really is the focus of where we are. So our muscarinic M1 program, for example, is driving on selectivity in an area that's been traditionally almost impossible to drive selective compounds for. I'd like to end with one final analogy. I think GPCRs will come back. I hope that this has provided a little bit more evidence that the way that we're approaching them gives you new opportunities for targets which are clinically validated but which have been very difficult or chemically intractable. So I know he probably didn't write the lyrics, but he did go to Manchester here. And I guess things probably will, or maybe in this case can only get better for him. Thank you very much.
Yeah, thanks very much, Alistair. I mean, that's a fascinating approach you're taking and really opening up the world of GPCR hit ID again, I think.